Hello everyone, and thanks for inviting me to present this talk here. I'm Gisela Gabernet. I work at the Quantitative Biology Center, QBIC, which is a bioinformatics core facility of the University of Tübingen. And as a core facility, Nextflow and NF-core are central for our analysis. We use NF-core pipelines for all the data analysis tasks at the core facility. And if we have to ever perform an analysis type that doesn't yet have a pipeline in NF-core or some functionality is missing, we contribute to NF-core to be able to share this functionality with the community. And today I'm going to talk about one of these pipelines, the NF-core airflow, that was for an analysis type that was not initially included in NF-core, so we went ahead and created a pipeline and shared it with the NF-core community. So what does the AIR in airflow stand for? AIR stands for Adaptive Immune Receptor Repertoire. And those are membrane proteins that are found at the surface of B cells, called B cell receptors, or BCRs. And they are also found at the surface of T cells, called T cell receptors, or TCRs. In the case of BCRs, when those are secreted in the media, they are also called antibodies, which is a term that we are all more familiar with. So the main function of all of these membrane receptors is to identify foreign antigens in the body. So for example, when there's a pathogen like a virus or bacteria, those receptors are meant to recognize this foreign antigen and induce an immune response. And so to be able to recognize this wide variety of antigens, we also need to have a wide variety of sequences in these BCR and TCR receptors. So it's estimated that in the human body, we can have 10 billion to 100 billion different receptors, BCRs and TCRs. So AIRR-seq is about getting the individual sequences of those BCR and TCR receptors that are present in an organism at a certain time. So this has an application in many different fields, ranging from studying the immune state of an individual at a certain time, but also detecting immune-related diseases where those receptors might not be functioning properly, guiding vaccine development, and guiding cancer immunotherapy. So I'm going to talk also a bit about the molecular biology of how the diversity of this TCR and BCR sequences is generated. In all cells, oh, sorry, those slides are not transitioning. Just realize now. Yeah, I can't explain that without a diagram. So, so in all cells of the human body, we have many different gene segments that are combined to generate these TCR and BCR receptors. Those are called the B, D, and J segments. In humans, we have 40 different B gene segments, 23 different D gene segments, and 6 different J gene segments and C segments as well. And what happens in these T cells and B cells is that there is a recombination at the DNA level. So the genome of these cells changes to pick just one of these each different gene segments and combine them into a functional receptor. And that doesn't really happen like Lego blocks, as you can imagine, but it's more like cut and paste procedure that is done by a 5-year-old that cannot follow exactly the lines. So there's also variability at the edges there, and some nucleotides can also be introduced. So there's variability in the sequences that is not really encoded in the genome even. In the case of B-cell receptors, there's even another process that generates even wider variety of these receptors upon antigen stimulation. So that's also what happened to all of us when we were first exposed to the coronavirus, or if we were lucky, when we received the vaccine to the coronavirus. So upon the B cells in the body, upon recognizing for the first step the antigen, they were activated, and they underwent a procedure of clonal expansion to generate children B cells, but all of them, they didn't have the same B cell receptor sequences as the progenitor cell, but they underwent a process that's called somatic hypermutation that introduces even more variability into this BDJ gene region. So all of these procedures basically generate a wide variety of BCR and TCR sequences, and it's estimated that together with this BDJ gene recombination, variable junction length, and in case of BCR somatic hypermutation, they are theoretically 10 to the power of 14 possible BCR sequences. So sequencing the AIR repertoires, there's several protocols available to do that, but most of them are based on amplicon sequencing, which is the targeted amplification of this BDJ-NC gene regions by library preparation and PCR, and further sequencing via several methods. And coming to the bioinformatic analysis, this one is not done like a traditional RNA-seq analysis because read mapping to the reference genome is quite challenging in that case due to the high diversity of PCR and TCR sequences, which is also not genome encoded, and also the genome region there, because of the BDJ repetitions, is highly repetitive. So there's the need for specific tools and specific reference data to do the bioinformatic analysis there. Luckily for us, though, we did not have to write those tools when implementing this pipeline. We came across the Immcantation framework, which is an open source toolset to analyze AIR-seq data that is developed at the Kleinstein lab in Yale. And there's a whole community of users already using this Immcantation framework tools. The developers were really helpful when we contacted them for the first time. So at this point, we decided to write a Nextflow pipeline, part of the NF-core community that uses the Immcantation framework tools, chains them together, and allows a high throughput, reproducible analysis end-to-end of this AIR-seq data. And we started at Cubic, developing this pipeline, but thanks to the NF-core community and the visibility that this gave us, we soon found collaborations across the world. First of all, with Susana Marquez, part of the Immcantation lab, which I met for the first time in the NF-core hackathon in person, so that was a great opportunity to meet the collaborators. And additionally, other contributors such as David Ladd from Monash University in Australia. So in a nutshell, what the pipeline does, it's able to process the AIR-seq data from start to end, both bulk and single-cell sequencing data. From the FASTQ files, it generates read assembly and quality control, correcting also the sequencing errors introduced during PCR and sequencing. It performs IgBlast to the specific reference data provided by the IMGT consortia that contains this PCR and TCR sequences. And further analysis steps include also identifying the sequences that belong to the same T-cell clones and B-cell clones, and reconstructing the lineage trees, giving rise to these B-cell tree sequences. As a final step, there are also several properties of the BCR and TCR repertoires that are computed, such as the abundance of the individual B-cell clones in the repertoire and their diversity. And also the QC of the different tools is collected in a multi-QC report. So here's an example on how the NF-core airflow report for specific data looks like. And there's, first of all, a collection of all the samples that are included in the analysis, the results of clonal abundance, clonal diversity, gene usage, and finally also the citations of all the tools that are used during the analysis. As mentioned also yesterday, it's very important to give credit to all of the original tool developers of the tools used in the computational workflows. So thanks to the NF-core community, developing workflows as part of the community, it's made easy in many ways. One of them is that the markdown documentation is automatically rendered on the NF-core website, which makes it very easy for pipeline users to find and follow. Here's the example of the NF-core airflow documentation, an introduction to what the pipeline does, a page providing all of the input and output options, all of the pipeline parameter options, and also NF-core provides a way of running pipeline tests on every release on AWS Cloud to have example results on the website, and also to ensure that all of the pipelines are portable to the cloud. And at this point, I would like to thank all of the collaborators that were also involved in the NF-core airflow development, Alex Belsa at Cubic Tübingen and Simon Heumers, and as mentioned also Susanna Marquez and David, which also joined the effort in developing NF-flow airflow. And thank you all for your attention.
Thank you, Gisela. That was a brilliant talk and great examples of, I mean, you're a member of the NF-core team yourself. You've been working with us for ages. So you're probably a little biased, but I think it's a great example of why it's such a great community to work with. We're running a little bit behind time, so I'm just going to do one question for you, if that's okay. Maxime asks, in which organisms can you use airflow?
So far, it supports human and mouse data, but the user can also provide their own reference data. So if they find or generate their own reference data for other organisms, that could be also possible to use.